Good day, everybody. This is Chris back again with the Ancient Scholar. I hope this video finds you all doing well, considering present situations and circumstances facing humanity. So today what I'd like to do is I'd like to continue on my COVID-19 / SARS-CoV-2 series of videos, and I'll be talking about some treatments. Specifically, I'm going to be talking about the development of vaccines and where we're currently at in contemporary vaccine development. This video will be fairly contemporary to 23 March 2020. So let's go ahead and get started. So to understand vaccine development, we have to understand a little bit about the immune system. Now, I actually have some comprehensive videos that I've done on the immune system earlier, and you're more than welcome to reference those, but I think today what I'll do is I'll shoot for more high-yield information, so we'll strip it down a little bit. So the basic situation is the following. When the body becomes invaded by a virus and that virus begins replicating, one of the first things that happens, aside from the inflammatory process, and aside from inflammation, is you have the frontline cells of the immune system known as the phagocytes. And phagocytosis just means to eat. And so what these phagocytes do is they engulf the virus and they eat the virus and they dump enzymes onto the virus, digest it, break it up. And these include white blood cells such as neutrophils, monocytes, and then when monocytes migrate out of the blood, they become macrophages, dendritic cells within your tissues. And so when these cells phagocytize the virus, destroy the virus, they also do something else. They find a component of the virus that can be used to identify the virus. And the interesting thing about viruses and bacteria, for that matter, is their cell walls have lots of different proteins in them, and these proteins can act kind of like a fingerprint, quote unquote, for a specific organism or a specific infective particle when we're talking about a virus. And one of the areas on a coronavirus that is very specific to the coronavirus are these spike proteins that we've talked about earlier. And so what these phagocytes can do is they can take that fingerprint, and that fingerprint is typically a protein of some sort, because proteins are reasonably large and fairly complex and diverse. And that component of the virus that can be used to identify it, its fingerprint, so to speak, is something known as an antigen. And so what these phagocytes can do is, in the process of phagocytosis, they can identify the antigenic component of the virus, and then they will present those antigens. So they'll find the antigens, and then they will present the antigens. And this is the reason why these particular cells are sometimes referred to as antigen-presenting cells, or APCs. And what will happen is these antigens will then get presented to another part of the immune system. Specifically, these will get presented to what are known as B lymphocytes. So the APCs, antigen-presenting cells, will present the antigens to the B lymphocytes, and that will allow the B lymphocyte to incorporate that antigen into another kind of molecule. And so here you can see on these little Y-shaped molecules here, these antigens have been incorporated into this open area. And these special molecules are often referred to as antibodies. And antibodies are also known as immunoglobulins, or Igs. Immunoglobulins, that means the same thing. And so the B lymphocytes mature, and they begin, once they are presented with the antigens, they can mature, and they will transition into something known as a plasma cell. And then these plasma cells can begin cranking out antibodies, And then because those antibodies have that antigen incorporated into them, they are very specific. They get pumped out into the bloodstream, and they can circulate around, and then they can identify the virus, bind to the virus, and deactivate the virus, and allow lots of other things to happen as well. They are what we call neutralizing antibodies. And then once the infection is over, the plasma cells can go into kind of a dormant state, if you will, and the plasma cells will quit actively producing antibodies, and they'll go in kind of a dormant state, and they will become what are known as memory cells. And then conceivably, the next time that particular person gets exposed to the virus, those memory cells, through a bunch of different mechanisms, can then be turned back into plasma cells and begin producing more antibodies. And this is where the term immunity comes from, when we say that you were immune to something. If you've had the infection in the past, there is a chance that you could have antibodies floating around in your body already produced, and you have these memory cells that can be turned back onto antibody-producing plasma cells. So that's kind of what happens there. And so what we can do is we can kind of make use of this system through something known as a vaccine. Because the way that this system works is you kind of have to get the infection to get the memory cells and the plasma cells and the antibodies, right? Well, if the infection's nasty and there are lots of comorbidities associated with it and even mortality, that's perhaps not a very safe option in some people. And so what we can do with the vaccines is there are several ways we can do it. One way we can do it is we can actually just take what's known as a live virus. So we'll take the virus, and this means that we have to grow it. And so we find tissues that it will grow in, animal tissues, for example. We'll culture the virus, we'll grow it, and then we can modify that virus to make it to where it does not cause disease, or it causes a very, very mild form of the disease. And this is sometimes referred to as an attenuated virus. So we're essentially taking the actual virus, attenuating it so it doesn't cause substantial disease, and then using it. And these are very effective, but there obviously are some risks as far as people can have a response and they can feel kind of poorly, so on and so forth. And an example of a very common attenuated vaccine that we run into is something called the MMR vaccine. So that's one way we can do it. Now, the problem with doing that with SARS-CoV-2 is, well, we've got to be able to grow the virus, and it's really nasty, right? The human immune system has never really come into contact with this, so it takes a lot. If we were to produce an attenuated vaccine for the SARS-CoV-2 virus, there are lots of safety measures that would have to be implemented for us to do this, right? What were called biosafety measures, and maybe I'll talk about the biosafety levels. But essentially, you're looking at a very high biosafety level. A lot of work would have to happen for us to develop an attenuated vaccine, and there's some risk associated with that as well. And it's not at all, at least I don't see an easy path forward that could get us a vaccine in any reasonable amount of time that way. Now, another way that we can produce a vaccine is through something called an inactive vaccine. So we take the virus, so we still have to culture the virus, we still have to grow the virus, but what we do is, instead of having a virus that's somewhat functional like we see with an attenuated vaccine, what we do is we add heat and other chemicals to the virus. And the heat and some of those other chemicals, enzymes, things of that nature, it's different for every virus or every vaccine. But what we do is we inactivate the virus, and the virus is incapable of producing disease following the treatment. And then that can also go in. Again, it's the antigen that we're most concerned about, getting that antigen to those phagocytes so we can produce antibodies. Now, the potential problem with an inactive vaccine is it doesn't stimulate as robust of a response. So it's not as robust of an immune response. So typically, the immunity doesn't last as long. And often these require boosters, so you have to every so often receive additional vaccines, and these are known as boosters. So an example of this might be something like the polio vaccine. The current polio vaccine might be an example of an inactive vaccine. Again, when it comes to developing a vaccine for SARS-CoV-2, there's a lot of work that would have to go into this. We have to grow and develop these viruses in tissue lines or cell lines, and again, the amount of time it would take is probably fairly prohibitive here, although there are definitely people working on this. Another kind of vaccine that we run into is something called a subunit vaccine. And the subunit vaccine essentially, where we don't, so these two vaccines here, the live vaccine and the inactive, the attenuated and the inactive vaccines, use an actual virus. Now, a subunit vaccine just uses a component of the virus. So maybe you grow the virus, and then you through different techniques can pull a certain component of the virus out and then concentrate that. So you use a component. Typically we pull a protein off of the virus, a protein that can act as an antigen. And then that protein can go on to elicit an immune response. This is, in fact, how the current hepatitis B vaccine works. All right. And then finally, the last major type of vaccine I want to talk about is something called I'll call these genetic vaccines. And I'll put quotes around that because that's just a vernacular term that I'm using. And so what we can do, so in live, inactive, and subunit vaccines, the attenuated and inactive, you're taking an actual virus, and in here you're taking a part of the virus. Now, the genetic quote-unquote vaccines are very different in that what we do is, instead of taking a part of the virus, what we do is we take the genetic code. We take the genetic code for a part, or the code for a component of a virus. And then we inject that genetic code into the person or animal, and then that person or animal will begin producing whatever component that genetic code is for. So they don't actually produce the virus, they just produce that component, and then the body might be able to recognize that component as an antigen and then produce the appropriate antibodies. All right. And we could either do this with DNA, right? We could have a DNA vaccine, deoxyribonucleic acid, or we could have an RNA vaccine, ribonucleic acid. And we attempted to do this, I believe, with the Zika virus some years back. A DNA vaccine was produced for the Zika virus, and unfortunately, that was an unsuccessful endeavor. So that's the unfortunate truth of all these is there's a lot of work that goes into developing vaccines, and there's no guarantee that any specific vaccine is going to be successful or work. Okay, so what I want to do is I want to talk about one of the vaccines that's being produced, or being developed rather. And it is really, the reason I'm talking about it is because it's the only one that's currently entering human trials. So there's a company known as Moderna. And they have actually kind of have specialized in producing these so-called genetic vaccines. And Moderna, back when the COVID-19 was an epidemic, China released the genome. They were able to get the genome, or the genetic sequence, for the entire virus, for the SARS-CoV-2 virus, and Moderna was able to identify a specific sequence of RNA that codes for a specific component of the coronavirus. And this is called messenger RNA, or mRNA 1273, mRNA 1273. All right. And what they did is they took that sequence, that RNA sequence, and that RNA sequence actually codes for a spike protein. So it codes for the spike protein that comes out of the viral envelope, and it is that spike protein that we think might be a substantial antigen and could induce an immune response. And then what they did is they took that and then they packaged that, or they encapsulated that in something called a lipid nanoparticle. A lipid nanoparticle. I believe they call it a lipid nanoparticle medium, or an LNP. Now, I don't know exactly what's going on there, but I suspect that surrounding or encapsulating the RNA in a lipid nanoparticle medium just allows that RNA to better get into cells, because cells have a lipid membrane, and so this lipid nanoparticle medium, small, it's lipids nonpolar, so it can much more effectively get into the cells, and then it can, once it gets into the cells, then it can cause cells to produce these spike proteins, and then that will go on to hopefully produce an immune response. That is my guess at this point. Obviously, Moderna is not, they haven't published all the details to their stuff, again, because, right, if they do, I would suspect there's a really good chance that somebody could copy all their work and produce their own vaccine, and then all that work will go to waste. And that's my assumption. I could be wrong, and feel free to correct me on that, but that's my assumption. So Moderna, in just unheard of, basically a very, very short amount of time, really unheard of to humanity, have produced a vaccine candidate. And so they have done some preclinical. So when we say preclinical, this is before this vaccine candidate is injected into human beings. So you got preclinical work. And typically what this involves is taking the vaccine or whatever medication you're developing and injecting it into animals such as mice, but not just any animal. What we do is with these animals, we have some control over the kinds of genes that these animals have, and we can take certain genes out and we can add certain genes to these animals. We call these knockout and knock-in animals, and we can make an animal more or less likely to react in a certain way. So we have a better understanding or a more reasonable idea of how human beings may react. And so you do some preclinical research, and then once you get the preclinical research out of the way and you have some reasonable expectation that this of what the safety parameters are going to be in human beings, then you can begin clinical trials on human beings. And to get a drug to the market, at least in the United States, following preclinical work, there are 3 phases of trials. So you have what are called phase 1 trials. And phase 1 trials where you give the substance, the vaccine in this case, to small numbers of healthy people. And essentially what you're trying to do is you're trying to determine what is the safety, right? What are the safety parameters? Is it actually safe to give to healthy people, right? That to say nothing of whether it works or not. So that's phase 1. And phase 1 can take a while. It can take months to years with some substances to really figure out what the safety parameters are. And then once we have some idea of that, then we can do what are called phase 2 clinical trials. And in phase 2 clinical trials, we give it to small numbers of at-risk people. So people that are at risk for a disease or people that may have the disease. And here what we're trying to figure out is there any effectiveness to it? Is it effective? And what's the dose, right? So is it safe? Is it effective? And then finally, once we have some idea of the safety and effectiveness, then what we need to do is we need to do large studies. And typically what we need to do is we need to have randomized studies, and we need to have placebos in place. So half of the people will get the drug and half of people will get a placebo, right? Because we need to make sure that this isn't a placebo effect, that this is an actual effect that we're accounting for. And these are referred to as randomized controlled trials, or RCTs. And typically that involves hundreds to thousands of people in different locations or different hospitals or different centers throughout the United States. And this is what we call a phase 3 trial. And then if it makes it through phase 3, then it can be approved by the Food and Drug Administration and go to the market. Now this typically takes, for something to work, for something fast, right? I mean, you're looking at 5 to maybe 10 or more years, right, to get a drug to the market. And currently, we suspect if everything works perfectly, we might be able to get this Moderna vaccine to the market in approximately 18 months. All right, so substantially faster. But again, that's if everything works out right. So where are we right now? Well, we've done some preclinical work, and we, in just the last couple of weeks, have begun phase 1 trials with this particular vaccine, with the mRNA 1273 in a lipid nanoparticle medium. I believe this is going in Washington State, and I believe that 45 healthy adults, both men and women, have been given or are being given this. And then this will last a few months, potentially several months. And we'll monitor these adults and see what kinds of side effects there are. And then in the next several months, if we have a good safety profile, then we can begin phase 2 trials, where we will begin administering this vaccine candidate to more people and figure out if it actually elicits an immune response and prevents infection and causes immunity in those small numbers of people. And then finally, we can begin phase 3 trials, where we can give this to hundreds, if not thousands, of people with randomization, with placebo, with blinding, and see how truly effective it is. And then hopefully, if all goes well, this might make it to the market within 18 months or so. So this is currently the best candidate for a vaccine that we have. It's ahead of all the other candidates, but there are certainly many other candidates that are being developed and might enter preclinical trials. Just be careful that when you look at the news and the news says that a new vaccine has been developed, really what they mean for the most part is that a new substance, a new vaccine candidate has been developed. And where is it in all this? Is it at the preclinical phase? Is it looking at phase 1 or phase 2 or phase 3, right? Because if something was just newly developed today, well, right, we need to do preclinical work on it first, right? It needs to go through the whole gauntlet. And currently, the only thing, the best that we've gone so far is phase 1, and we have no data from phase 1, so we don't even know if it's safe, let alone effective in large numbers of people. We just don't know that yet. So there is still a lot of work to do, and there is no guarantee that this messenger RNA vaccine will even be effective. Hopefully it is, however. Okay, hopefully that makes sense, and hopefully you understand where we're at in vaccine development a little better. Take care, everyone.